The next component of bacterial cell is capsule, the outermost layer that is found around the cell wall of bacterial cell. In general, we call it as glycocalyx. So, it is not that all bacterial cells possess this glycocalyx, but there are few bacteria which possess this bacterial glycocalyx. These bacteria they produce viscous material outside their cell wall, and that is called as glycocalyx. In general, it is made up with carbohydrates, and therefore the reason why it is called as glyco. This glycocalyx it is composed of monomers. That means it is a type of polymer, and it is made up with hundreds or thousands of repeating units of monomers. So this glycocalyx it may be made up with carbohydrates majoritarily. It may be made up with protein in some cases, and in some cases it may be made up with glycoproteins. So this glycocalyx in general is divided into two categories. First one is capsule. So in general what we can see here is this is the bacterial cell with bacterial cell wall, and around which we can see one more organized layer, a well organized layer, or which is in particular shape. This organized layer of glycocalyx it is called as capsule. This means that this layer is firmly attached to bacterial cell, and it is made up with polymer, and that polymer is regular, or it is organized in a proper fashion or intact manner. So when the glycocalyx is organized in a defined structure attached firmly to the cell wall, it is called as capsule. Whereas sometimes we find that this glycocalyx is disorganized. That means it does not have a specific definite shape outside the cell wall, and it is also not firmly attached. It is loosely bound to the cell wall. That means it can be released. It is not firmly bound, and sometimes it is soluble in water, and it is responsible for increasing the viscosity of water. This is called as slime, bacterial slime layer, which is like this. So it is loosely bound. We can see the structure, and it may be released if it is present, or it is shaded if it is present in aqueous environment. So like this, such type of bacteria are responsible for clogging of for example pipes. So this is the difference that you can see between a bacterial capsule and the slime layer. This is how we can see it under a light microscope with a magnification of 1000 or 1500x. So simple microscope. This is a negative staining technique in which we can see at the center there is bacterial cell, and around which this colorless part, this is bacterial capsule which is made up with polysaccharide, and this is the background which is dark. So we can see the background is colored, cell is colored, and this capsule is colorless, and that is why we are able to see it. This is the staining technique which is generally used in microbiology laboratory. We can see that a chain of bacterial cell which are bound to each other and which is colored in blue. The background is dark and the capsule, it remains colorless. So in this case capsule is not stained, and that is why it is called as negative staining for the demonstration of capsule. This method is Manavel's technique for the demonstration of bacterial capsule, which you must have learned during your practicals. The composition of capsule, in most of the cases it is made up with polysaccharide. This polysaccharide capsular material, it can be of two types: homopolysaccharide capsule. We know that polysaccharide is a polymer that composed of monomers or sugars or monosaccharides. When these monosaccharides are of same type, so the repeating units of same type of monosaccharide is there, then it is called as homopolysaccharide. So it is made up with single kind of sugar or monosaccharide. For example, the capsule of Streptococcus mutans. This is the name of a bacterium. It is composed of a polysaccharide material called as glucan. So this glucan is a type of homopolysaccharide and which is made up with repeating units of glucose. So the use of this glucan is that with the help of this bacterium, it can bind to firmly, it can bind to the smooth surfaces like our teeth, and because of that, if it persists there, it can populate on the surfaces of teeth, that leads to the development of dental plaque, and further it leads to dental caries or cavities. Another type of capsular material is opposite to that, it is heteropolysaccharide. Hetero means more than one type. This type of capsule is present in case of Streptococcus pneumoniae, which is also a type of pathogen like Streptococcus mutans. So this capsular material, it consists of the repeating units of monosaccharides such as glucose, galactose and rhamnose. So in contrast to this is composed of only glucose, but this is composed of more than one type of monosaccharide and which are repeatedly present: galactose, glucose and rhamnose, and that is why it is called as heteropolysaccharide. There are different types of bacterial pathogens, animal pathogens or human pathogens, which consist of these types of capsules, but there may be differences in the composition of sugar combinations. So presence of capsule, it is one of the characteristic of many of the pathogenic bacteria, because with the help of capsule these bacteria can survive in our body, and this capsule, it helps these bacteria to establish their population in a particular location or in body tissues, that we'll see under the functions of capsule. So the capsule of some bacteria, it may be made up with proteins or polypeptides and not polysaccharide. This type of capsule is seen in case of Bacillus anthracis, which is a causative agent of anthrax disease of animals, and it is a polymer of amino acid called as glutamic acid. Now, the functions of glycocalyx as well as the importance of glycocalyx. Function and importance in relation to its positive effect and negative effect. Now this glycocalyx, it can serve a number of functions depending on its presence in bacterial species. First function is it helps in the binding or attachment. So if bacterial cell has this capsule, it can bind to the several solid substrates, for example, plant roots, human teeth and tissues, rocks in the faster moving water. The causative agent of dental caries is Streptococcus mutans, and this Streptococcus mutans, it remains bound to our teeth with the help of the sticky capsule and gets protection from being washed away by the flushing action of saliva, and this permits the start of the infection with dental plaque, and further it progresses to dental caries, and this is how the development of dental plaque, and further it leads to development of dental caries. Second function of capsule is protection. It protects or it gives protection to bacterial cell from dehydration conditions. That is, it can protect the cell against temporary drying and desiccation, because this capsular material has ability of binding to water molecules. Then third function is again protection. So presence of capsule ensures the protection of bacterial cells from getting infected by viruses. The viruses of bacteria, they are called as bacteriophages. So if bacterial cell is having capsule, then these bacteriophages cannot bind to the bacterial cells, and therefore if it is not able to bind, therefore there will be no infection to bacterial cell. If there is no infection, there will be no destruction of bacterial cell. So this is how capsule may protect bacterial cells from getting infected by viruses. In some cases, as capsular material is made up with polymer, it may serve as a stored food or source of energy. Again, the capsules protect bacteria from the phagocytic cells which are present in our body, in mammalian's body. So, capsule protect bacteria from being phagocytosed or engulfed and digested by white blood cells, which are called as phagocytic cells, which we have learned in the last semester. So therefore, if bacterial cell is having a capsule, then it escapes from phagocytic cells, and therefore it gets an extra life, it gets a protection from phagocytic cells, and that permits the start of infection, and that leads to the production of disease. This is how the diagram represents this particular function or importance of capsule. So for example, this is phagocytic cells and this is bacterial cell. So phagocytic cell, it engulfs this bacterial cell and it digests it. But if this bacterial cell contains this capsule, so the first step in this is the binding. This phagocytic cell, it binds to the bacterial cell. But because of the presence of capsule, this phagocytic cell cannot bind to this bacterial cell. So the binding is inhibited because of the presence of capsule, and therefore the bacteria cannot be engulfed. So this binding is blocked, and therefore this bacterial cells remains free and they can cause infection. So phagocytic cells, we have learned that they are involved in second line of defense. So this is how bacterial cells, they get escape from second line of defense mechanism. So these glycosylated bacteria, they can cause severe problems in industries also. This is the importance, means it is showing the bad effects of capsule for the human welfare. For example, in industries, they are responsible for the accumulation of slime. So they secrete slime which is loosely bound. So this slime is accumulated in the manufacturing equipments, and that can cause the clogging, or that can cause the blocking of filters, blocking of pipes, and non-functioning of different types of equipments, therefore decreasing the quality of final product. So presence of slime or production of slime by bacterial cell, it causes the increase in the viscosity of aqueous liquids, and that causes or that affects the flow of that aqueous liquid because it increases the viscosity.